Hello everyone, so this is the GCSE Combined Science Trilogy walkthrough for Foundation tier, and this is Biology Paper 1 that was sat by pupils in May 2019. So I'm going to go through and explain how I would have tackled this paper. I've also put links to the blank paper and mark scheme in the description, along with question by question revision videos if you need to go back and revise the content. So, question number 1 then. A student investigated the effect of light intensity on the rate of photosynthesis, and it shows some of the apparatus used. So they've got a stopwatch, they've got their pondweed in a beaker in water, and they've got bubbles of gas being produced. Name the gas produced by the pondweed in the light. So this is linking back to our photosynthesis equation. We should know that plants take in and absorb CO2 and produce oxygen. So this whole required practical is about counting the amount of oxygen bubbles produced in a certain amount of time. Describe one way the student could change the light intensity of the light reaching the pondweed. Well, the other piece of equipment that you tend to have in this practical is a lamp that is shining the light. How can you adjust the lamp's light intensity? Well, you can change the distance of the lamp. You can either have it really close or you can move it further away, and that will adjust the light intensity. So 2 marks here. You could use a lamp, and then you could change the distance between the lamp and the pondweed. So question 1.3 then. Describe how the student could use the apparatus to measure the rate of photosynthesis. So when you're talking about the rate, you're talking about how fast it's going. But in order to calculate rate, you need to know how fast it's happening in a certain amount of time. And it's this part that people often miss off. So what you should have done when you did this required practical was you could count the number of oxygen bubbles made. That would get you 1 mark, but then you also need to say in a certain amount of time. Now you could make it up. It hasn't given you any limitations here, so you could say 30 seconds, a minute, whatever seems logical to you. Figure 2 then shows the student's results. You've got the light intensity increasing on your x-axis, and you've got your rate of photosynthesis on your y-axis. What is the maximum rate of photosynthesis? So we use the graph and we see, well, where does it actually go up to? Okay, and we can read off our graph. Obviously, you would use a ruler in the exam, and you can see it goes pretty much up to 34 arbitrary units, and then it levels off. Question 1.5. At which light intensity was light a limiting factor? 200, 600 or 1200? So if we go back to the graph, we're looking for where light is having a direct impact on the amount of photosynthesis. So at 1200, we can see that it's leveling off, it's leveled off, and it's not going up anymore. So 1200, light intensity at 1200 is not a limiting factor. It's not slowing down the rate of photosynthesis. Again, at 600, the line is flat again, is where it started to level off. So therefore, it's got all the light that the plant needs. It doesn't matter if you add more to it, you're not going to get more photosynthesis. So therefore, it must be something else that is slowing down the rate of photosynthesis between 6 and 1200. However, at 200, you can see light is having a direct impact. If you have more light, you have more photosynthesis. So therefore, we would say it's at 200 that light is a limiting factor. So light intensity can affect the rate of photosynthesis. Give one other factor that can affect the rate. So you've really just got to think about what else do plants need? Well, think back to your photosynthesis equation. CO2 plus water makes glucose, C6H12O6, and oxygen. What else do plants need then? Well, they also remember, they need the light energy and they need chlorophyll. So the other things you could say that are your limiting factors would be carbon dioxide concentration, okay? You would say the amount of chlorophyll. You could also say temperature, and you can also in this one say the amount of water available, because it would affect the rate of photosynthesis. So just think of the other things that plants need. Question 2 then. A single-celled organism has a large surface area to volume ratio. How does oxygen enter a single-celled organism? Well, oxygen is a particle. How do particles get into our cells? It should be diffusion, because osmosis is just to do with water, and active transport, well, I'm sure we'll come on to that part in a bit. Figure 3 shows two cubes. The surface area the surface area to volume ratio for cube A is 6:1. Calculate the surface area to volume ratio of cube B. You've been given the measurements of cube B, and this is worth 4 marks, and you can kind of see the question, how they've laid it out, is guiding you through how you could go about calculating it. So it says, the first thing you need to do is calculate the surface area of one face of cube B. So you're just calculating the area of one face of it. Now you've been given the measurements. Remember, area is just base times height, so you know it is just 2 times 2, which would equal 4. Then you would then have to calculate the total surface area of cube B. So the total surface area of cube B, you need to think about how many sides does a cube have. Well, it has its 4 sides going around the outside, and it has its top and bottom. So in total, it has 6 sides. So you would do 6 times by the area of one side, which is 4. So 6 times 4 is 6, 12, 18, 24. You then need to calculate the volume of cube B. So you should remember from maths that volume is base times height times width. So this one you would just do 2 times 2 times 2, which gives you 8. So your surface area to volume ratio, you calculate your surface area there, which is 24, and then you've got your volume here, which is 3. Okay, so your ratio, 8. So therefore, your ratio is simply 24 to 8. You can simplify it if you want to, but you don't. It hasn't asked you to do it. So you can get a way of simplifying it, dividing it both by 8, because 24 is a multiple of 8, and just put it as 3:1. But if you left it at 24 to 8, that would still get you the 3 marks. Multicellular organisms have exchange surfaces to absorb substances. Figure 4 shows part of the exchange surface in the lungs. Question 2.3. Oxygen passes from the alveolus into the blood. Name the part of the blood that carries the most oxygen. So remember, in blood there are 4 parts to it. You've got your red blood cells, you've got your white blood cells, you've got your platelets, and you've got your plasma. Now the part that carries oxygen, you should remember, is your red blood cells. White blood cells fight disease. Platelets, they cause clotting and form scabs, and plasma carries everything else, like hormones and CO2. Name the gas that passes out of the blood in the alveolus. So if oxygen's going in, you then have carbon dioxide diffusing out, because that's the gas that we produce from respiration and we want to get rid of. Question 2.5. The alveoli provide a large surface area for gas exchange. Give 2 other ways the lungs are adapted for good gas exchange. Now this is a common question. They can relate it to any situation. They might talk about the alveoli, the intestines, adaptations of fish, but you can say the same adaptations if it's talking about gas exchange or movement of particles. Okay, so how are the alveoli adapted? Well, you can't use large surface area, but you can say they've got thin walls, so there's a short distance for the particles to diffuse. So that means it would happen quicker. And then you could also say it has a good blood supply. So there's constantly new blood being pumped around the alveoli, and that makes it a steeper concentration gradient, so therefore the particles move quicker. So generally your three adaptations of gas exchange are large surface area, thin walls, good blood supply. Question 3 then. This question is about the organisation of living organisms. Write the biological structures in the box in the correct size order, starting with the smallest. Well, we should know that out of these four, the one that is inside the cell would be the nucleus. Then the cell is what contains the nucleus. A group of cells working together is a tissue, and a group of tissues working together makes an organ. Okay, so the definition of a tissue is a group of similar cells working together to do a specific job, and a definition of an organ is a group of tissues working together to do a specific job. Now the next one, name one animal organ, possibly the easiest question in the paper. You just need to name one organ in your body, 1 mark, don't overthink it. So you could say any from this list: bladder, brain, heart, small or large intestine, kidney, liver, lung, pancreas, skin or stomach. So any of those, just have to put one of them and that would give you the mark. Question 3.3 then, which is a plant tissue? So you've got the flower, the leaf, the phloem and the root. Now, the flower, leaf and root, these are quite big structures in a plant. Because they're quite big, they have main functions. They're made of many tissues, so these three are plant organs. Whereas the phloem is one group of cells that work together to do a job. This one moves sugar up and down the plant, so therefore it is the only tissue. Figure 5 shows part of a root viewed from a microscope. Okay, so the image that was here, we can't display, but basically it just showed a cell looking a bit like this. Okay, this is a root hair cell. So you've got a cell that has a thin extension. Explain how a root hair cell is specialized for its function. Well, we should know that the root hair cell, its job is to get water and minerals for the plant. How is it adapted? Well, it's similar to the question on the alveoli before. The thin extension gives it a large surface area, and it also has a large surface area, so it can absorb a lot of water and minerals. You could also say that it has many mitochondria to allow it to do active transport, but that is very specific knowledge. Figure 6 then shows a microscope. You've got the eyepiece lens, you've got the objective lenses, you've got a stage, and you've got the prepared slide. Question 3.5 says it is easier to view cells using the low-powered objective lens first. So you can see they've started with the smaller one. Give one reason why. Okay, why is it easier to view cells using the low-powered lens? Well, it's simply because the easiest answer is that it's easier to focus. Okay, it's a low-powered lens, therefore it's easier to focus, and it also gives you the wider view of the sample. Think of it like when you take a picture, you always if you take a picture of your friends, you always scan the whole picture to begin with and check it what it looks like, and then you zoom in and look at your specific face, or you look at your friend's face. Okay, so you start with the lowest lens and then you zoom in on the part you then want to look at. Question 3.6 then. To focus the image, the objective lens should be moved away from the stage. Okay, give one reason why the objective lens should not be moved towards the stage. So why should we never, when we use a microscope, push or move our objective lens downwards? Well, if we keep on moving it downwards, the lens and then the slide would then hit each other and they could crack. So the lens or the slide could collide and break, which is obviously not what we want. Question 3.7 then. The image of the prepared slide in figure 6 is viewed with the ×10 objective lens. The total magnification is ×50. What is the power of the eyepiece lens used? So this is when you've got to know the equation for magnification of a microscope. The total magnification of a microscope is equal to the eyepiece lens times the objective lens. Now, if it's telling you the total magnification is 50 and the objective lens is 10, what must the eyepiece lens be to make 50? Well, it would be 5 times 10 equals 50. Question 3.8. Root hair cells do not contain chloroplasts. Suggest a reason why. Well, if you think about the roots, they are in the soil, and the job of the chloroplasts is for photosynthesis, and they absorb the light. So why do root hair cells not have chloroplasts? Well, it's because they are in the soil, then there is no light. Okay. Question 4 then. Water is lost through the leaves of a plant through stomata. Which is the process by which water is lost from the leaves of a plant? Now, water being lost out of the leaf, okay, is called transpiration. The movement of water from the roots going up the stem out of the leaves is called the transpiration stream. Osmosis is the movement of water in and out of cells across the membrane. And obviously photosynthesis is how plants make their food. Question 4.2. What is the name of cell X? Well, if we look back at the picture, the stomata are the actual holes, okay? But they're surrounded by two cells which can open and close. And it's either epidermal guard, palisade or xylem. And it is the guard cell. So think of it, they are guarding the entrance to the cell or to the plant. Question 4.3. The length of a cell is 25 millimeters when viewed at a magnification of ×800. Calculate the real length of the cell, give your answer in micrometers. 1 millimeter is 1,000 micrometers, and they've kindly given us the equation. The real length is size of the image divided by magnification. So we can substitute in our values. The length of a cell, so the size of the image, is 25. Divided by the magnification, which is 800. But we can't just do 25 millimeters because it asked for it in micrometers. So we need to convert 25 millimeters into micrometers. But it tells us there's 1,000 micrometers in a millimeter, and I've got 25 millimeters. So therefore, I would times it by 1,000 to give me 25,000 micrometers, divide it by the magnification 800, and that gives us a reading of 31.25 micrometers. Then, question 4.4. We've been given a method for a little practical. A student measured the mass of water lost from some similar plants. The plants were at different temperatures. Figure 8 shows the apparatus used. So you've got a scale, you've got a plant and a plastic bag around the base of the plant and in its pot. So this is the method they used. They sealed a plastic bag around the pot of a plotted plant. They put the plant with the bag on a balance in a room at 20 degrees. They recorded the mass of it. After 3 hours, they recorded the mass again, and they calculated the amount of water lost from the plant. They repeated steps 1 to 5 at 25 and at 30 degrees with similar plants. So what was the independent variable in the investigation? The independent variable is what they change. So therefore, what are they changing in this method? Well, we can see from point 6, they're doing it again at different temperatures. So out of the options, they're not changing the initial mass of the plant, they're not changing the length of time it was left, they're measuring the mass of water lost, but they are changing the temperature that the plant is going into. Suggest why the student placed a plastic bag around the pot. So you're measuring how much water should be coming out of the leaves through transpiration. What we don't want to happen is have water evaporate out of the soil that never went into the plant to begin with. So the whole point of the plastic bag is to trap water inside the plant and the pot, and to ensure water is only lost from the leaves. Table 1 shows their results then. You've got the temperature at 20 degrees, 25 and 30 degrees. You've got the mass at the start, the mass after 3 hours, and the mass of water lost in 3 hours. What is the resolution of the balance used in the investigation? Well, the resolution of a device is the smallest change a device can measure. So if we look at these options then, we can see that each of these go to 1 decimal place. So therefore we would look at which of these boxes goes to 1 decimal place, and it's just the first one, so it's 0.1 grams. So it's 510.7 or it's 510.8. The smallest change it can measure is 0.1. Calculate value X in table 1. So we're calculating the mass of the plant at the start. So it's finished up with 506.3 and it's lost 4.9. So all we would do to calculate the initial mass is add the amount of water we lost back onto it. So if we add those numbers together, 506.3 plus 4.9, that gives us 511.2 grams. Give one conclusion that could be made from the results in table 1. So it's just a 1 marker, so we can just state the overall pattern. Look at what we're changing, the temperature, look at the mass of water lost, where we can see as the temperature increases, the mass of water lost increases. Easy as that. Question 4.9 then. Give 2 factors that might affect the rate of water loss from the leaves, but we can't talk about temperature. So what other things might cause a plant to lose more water? Well, it could just simply be, going back to the first practical, light intensity. If you have more light, it's going to lose more water. And also wind or air movement. That would move more water out of the plant. And you could also then say as well, rate of photosynthesis, because if more photosynthesis is happening, then the plant is going to be using up the water and therefore it will lose more. Question 5 then. Malaria is a disease transmitted by mosquitoes. Figure 9 shows information about the number of deaths from malaria. calculate the decrease in the number of deaths between 2000 and 2015. 2 marks. So this is when it comes important to read the graph axes carefully. So you've got the year, but it's the number of deaths from malaria in thousands. So this would be, if I was reading at 400, it's actually 400,000 people. Okay, so that will be important when we do our calculation. So at 2000, okay, if we're calculating the decrease in the number of deaths between 2000 and 2015, if we read off our scale, in 2000 it was 840, but in 2015, if we read along, okay, just below 450, so it's 440. So we would then do 840, but remember it's 1,000, minus 440, remembering it's 440,000 again, and therefore the difference is 400,000. Okay, so question 5.2 then. Which time period shows the greatest decrease in the number of deaths? Well, if we go back to our graph, so which 5-year period shows the greatest decrease? Well, it would be the part of the line where it goes down steeper, so it would be between 2005 and 2010. A student looked at figure 9 and concluded that there were 800,000 deaths from malaria in 2002. Suggest one reason why this conclusion might not be correct. Well, if we go back to the graph and go to 2002, we can see it does go to 800,000. However, why might not this be true? If we look at the graph, there was no actual data collected in 2002. It's just on the line of best fit. So therefore it is an estimate. So it's no data collected in 2002. It was just an estimate using the line of best fit. Or you could say that another reason it might not be correct is that not all deaths were recorded. Okay. What type of pathogen causes malaria? Well, malaria, this is just a fact you need to know, is caused by a protist, and it's obviously spread by mosquitoes, which we call a vector. A vector is an animal or an organism that spreads a disease that is not affected by it. Question 5. Scientists are now developing a vaccine against malaria. Suggest how a vaccine against malaria could reduce the spread of the disease. Well, a bit like with COVID, if we're all vaccinated against it, it makes people immune. So people being vaccinated makes people immune, so they do not develop to the disease. So therefore, fewer people are then infected as the disease is passed on. Question 5.6. Give one way of controlling the spread of malaria, but we can't refer to a vaccine. So what we effectively want to do then, if you want to control malaria, is stop the mosquitoes from biting you. We want to get rid of the mosquitoes. So what we can do, if you live in a country that has mosquitoes, you can sleep under nets. Okay. You could then, to actually get rid of the mosquitoes for good, you can use insecticides, which are chemicals which would kill the mosquitoes. You could use insect repellents. Or you could then actually kill the habitats or destroy the habitats of the mosquito. If you destroy the swamps of where they actually live, then actually they won't be able to breed, and therefore they won't be about to infect people. Question 6 then. Figure 10 shows a human heart. Which blood vessel carries deoxygenated blood away from the heart to the lungs? So you've got your four chambers of the heart here with your blood vessels going out A, B, C and D. So a quick recap. Blood enters the right atrium from the vena cava, which is your main vein, bringing blood back towards the heart. It then goes through the right atrium and right ventricle. It then needs to go off to the lungs to collect oxygen, and B, the blood vessel, is the pulmonary artery, because arteries always carry blood away from the heart. When it's got its fresh oxygen from the lungs, it will come back to the left side of the heart through the pulmonary vein, and then it is pumped off again through the main artery called the aorta. Now, with the key thing to note from this is that the arteries, the aorta and the pulmonary artery, carry blood away. Just remember, arteries go away. And then the veins, the vena cava and the pulmonary vein, carry blood towards the heart. So arteries carry blood away, veins carry it towards. But which one carries deoxygenated blood to the lungs? It would be B, because that's pumping the blood to the lungs to get the oxygen. The natural resting heart rate is controlled by a group of cells that act as a pacemaker. Where in the heart are the pacemaker cells found? Well, the pacemaker, it's just a fact you need to know, is in the right atrium, and that just makes sure that your right side of the heart and the left side of the heart beats at the same time, so it releases the blood in a coordinated way. Some people may be treated with a drug to slow their heart rate. Digitalis is a drug that slows the heart rate. Where does the drug Digitalis originate from? Okay, now there are two drugs that you need to know. Digitalis originates from the plant foxgloves, and aspirin originates from the plant tree willow. Okay, so bacteria hasn't originated any drugs. Okay, a mold was actually used to create the first antibiotic, penicillin, and willow is used to make aspirin. Beta blockers are another type of drug that slows the heart rate. Table 2 shows information for people who do not take beta blockers and for people who do take beta blockers. Stroke volume is the volume of blood pumped out of the heart each time it beats. Cardiac output is the total volume of blood pumped out of the heart each minute. So you've got the table there, and you've got comparing people that have taken not taken beta blockers and taking beta blockers, and you've got the different volumes of at rest and during exercise for heart rate, stroke volume and cardiac output. You've been given the definitions above, and it then says calculate the stroke volume in table volume X in table 2, and you've been given the equation here. But we want to calculate the stroke volume, so you have to rearrange the equation. So you would move heart rate, divide by heart rate, both sides of the equation. So stroke volume would equal cardiac output divided by heart rate. You would then get these two values. Okay, so your cardiac output is 2,800. Your heart rate is 52. Just getting these numbers from the table there, divide them, and that gives you 54 centimetre cubed. Question 6.5 then. It says some people who take beta blockers often get out of breath when they exercise. Explain why beta blockers can have this effect during exercise, referring to information from table 2. Well, we can see clearly the effect of taking beta blockers is that it reduces your heart rate, that's lower. It then reduces your stroke volume, and then it reduces your cardiac output. So taking beta blockers is reducing your heart rate, the amount of blood pumped in your stroke volume, and therefore the total amount of blood pumped out each minute. So it has negative effects on your blood flow. So why might that affect people and they get out of breath when they exercise? So we can break it down into to do with exercise then. So when you exercise, you're obviously going to need more oxygen for respiration. Beta blockers reduce blood flow. And then you could quote data here from the table for heart rate, cardiac output, and stroke volume. So literally just copy the numbers, quote people that don't take it compared to people that do. Now, because they've got reduced blood flow, they would have less oxygen to the muscles. So therefore, if they've got less oxygen to the muscles, there would be less respiration and less energy released. So therefore, it would keep on, the muscles would get tired quicker. You could then link it again to the breathing rate. If your breathing rate is slower, you would have less oxygen being absorbed into the blood. Okay, you would also then have more CO2 in the body, which we don't want and doesn't help us exercise. Okay, yeah, and effectively, if you just say these key points to do with respiration, and the key thing is quoting loads of data, that will get you the 6 marks. Question 7 then. This question is about digestion. Name the enzyme that digests starch in the human digestive system. Now, starch is a type of carbohydrate. So remember, all enzymes end in A's. So the name of the enzyme is carbohydrase. The specific example that is found in your saliva is called amylase. So you can get the mark if you give the specific name of it in the saliva, amylase enzyme. A student set up a model to represent the digestion and the absorption of food molecules in the digestive system. Figure 11 shows the student's model. You've got a water bath at 37 degrees. You've got a partially permeable tubing inside a test tube. And inside that tubing, you've got starch and amylase solution. You've got the water and test tube. So this is their method. They fill the test tube with water at 37 degrees and they test it straight away. They test it straight away for starch and sugar. They mix together the starch and enzyme solution and immediately test it for starch. They fill some partially permeable tubing with the starch and enzyme mixture. They seal the tubing and place it into the test tube of water. They place the test tube in a water bath at 37 degrees, and after 30 minutes, they test the mixture inside the partially permeable tubing and test the water in the test tube for starch and sugar. So breaking that quite complex method down, effectively, they've set up this equipment. They have got starch and sugar in here. Okay, they test it at the beginning when it first get mixed together at 0 seconds, and then they leave it for 30 minutes and they test it inside and outside after 30 minutes. Okay, so it's before and after. Suggest which parts of the body the partially permeable tubing and the water in the test tube represent. So the partially permeable tubing, this is to do with absorption. Okay. Now, the part in of our body that does the absorbing of food nutrients, the organ, is the small intestine. So this partially permeable tubing is representing the small intestine. The water in the test tube is representing where the food nutrients go after it's been absorbed through the small intestine, and that is into the bloodstream. Okay, that then goes round to your different cells, delivering the glucose, etc., that your cells need. Table 3 shows the results, and they've tested it, the mixture inside the tubing at the start and the water just outside of the tubing at the start. They've tested it then again after 30 minutes inside the tubing and in the test tube after 30 minutes. And you've tested it for starch and sugar. It then says name the reagents used to test for starch and sugar. Reagents, just a fancy word for meaning what chemical do you add to it. Now, testing the starch, this is just your food tests, your food test required practical. So starch, we use iodine, which is the orange solution. And then for sugar, we use the blue solution, Benedict's. Question 7.4 says, why was there no sugar present in test 1? So in test 1, is at the beginning, it's inside the tubing at the beginning, and there is plenty of starch, but there's no sugar. We should remember what the enzyme is going to be doing. If this is our starch molecule, which is a complex carbohydrate, the amylase enzyme is going to come along and it's going to break the bonds between it and then make it into sugar molecules, simple sugars. Remember, an enzyme breaks the food down, so it's chopping up the bonds between it. So why was there no sugar present in test 1? Well, it's at the very start. If you've literally just mixed it together, okay, the enzyme hasn't had a chance to break it down yet. So there's no sugar as the enzyme has not had time to break it down. Question 7.5 says, explain the results for test 3. Well, inside test 3 then, you've got the mixture inside the tubing after 30 minutes. So again, we're testing what's inside the tubing of starch and amylase. And after 30 minutes, you've got starch and amylase, 2 marks. So we now have sugar, okay, because we can say the starch has been broken down into sugar. There is starch, as now the enzyme has broken down starch to sugar. However, as there is still starch, it's showing that the enzyme is not done. It hasn't finished digesting everything. So, however, the enzyme has not finished, and starch is still present. Last question on this paper then says, explain the results for test 4. So, test 4, if we go back and look at the table, it's saying for water, we are testing the water in the test tube. So outside of the tubing, it's diffused out after 30 minutes. There's no starch, but there is glucose. Now, if we think back to our partially permeable tubing, like our intestines, allows small molecules through, like glucose, but not starch. Okay, so we can link that into our question now. So why was there glucose? So we have glucose, but there is no starch. Well, that is simply because glucose is smaller and can diffuse, keyword, if substances travel by diffusion, can diffuse through the membrane. But starch is much bigger and cannot diffuse through. It cannot get through that partially permeable tubing. Okay, so that is the end of the paper then. I hope it was useful. Remember, on the link on the right, you've got a copy to the blank past paper and the mark scheme, along with revision videos tackling each question if you need to go back and revise the content for it. I hope it was useful, and of course, have a look at the other videos if you found it useful. Thank you.